Hi, Miss Nikki here. Welcome to Chapter 10, Part 2. We're going to cover the 4 phases of muscle contraction. So the first phase we have is excitation. So what are we trying to excite, right? We're trying to excite the muscle. You might have heard me talk about turning a neuron message into a muscle message. That's what we're doing in excitation. Then we're going to have excitation-contraction coupling. So how do I turn this new message? I have a muscle message, but how do I get the muscle to contract? So this is muscle message to contraction. Remember, we have to go from the outer sarcolemma and we have to get that message really deep to the interior where the sarcomere is located in order for the muscle to be told to contract. So it's kind of, we now have the muscle message. How do we get it to the core of the muscle where we can actually have contraction occur? And then 3, we actually have contraction. And this is where we're going to see the myosin from the thick myofilament and the actin from the thin myofilament. They're actually going to join together. We're going to have contraction or pulling or a shortening of the sarcomere from Z disc to Z disc. And then the last phase is relaxation. The muscle has to go back to its resting membrane potential so that we can do all of this activity over again. We've seen this image before, but let's kind of break it down and go through it. We know that there's a motor neuron. So this is a neuron message, right? We know that skeletal muscle is voluntary. So where's this message coming from? Right? Your brain is sending a message down a neuron to this neuromuscular junction. And here we have our muscle, our skeletal muscle. So this arrow is supposed to be showing you a message, a nerve signal. We're going to start calling that an action potential. Potential. There we go. So when this neuron message comes to the end of the neuron, the foot of the neuron, which is the synaptic knob, that action potential causes these calcium channels to open. When the calcium channels open, calcium from outside, so this is extracellular calcium, is going to flood into the synaptic knob. When the calcium floods into the synaptic knob of the motor neuron, it causes the vesicles inside the motor neuron to release acetylcholine. The acetylcholine diffuses into the synaptic cleft, and the acetylcholine will bind acetylcholine receptors on the sarcolemma or the plasma membrane of the muscle fiber cell. So here's kind of the wording behind what we just looked at. We know that calcium is going to enter the synaptic knob of the motor neuron. We know that that calcium is going to bind to proteins on the synaptic vesicles, and they're filled with acetylcholine. The vesicles are going to merge with the cell membrane and get dumped into the synaptic cleft. Thousands of acetylcholine molecules are going to be released into that synaptic cleft, and they're going to bind acetylcholine receptors on the muscle. So basically, we've passed the baton or the relay. We've turned a neuron message into a muscle message. Let's go through this image one more time. Here's our motor neuron, and we can see that there's an electrical message being sent down the axon to the synaptic knob. And this electrical message we also call an action potential. And this electrical message, when it reaches the synaptic knob, it causes these voltage-gated calcium channels to open. The calcium floods into the synaptic knob. The calcium binds to proteins on the vesicles, and the vesicles are filled with acetylcholine. When the calcium binds, the acetylcholine is released, and it's released here into the synaptic cleft, and eventually the acetylcholine is going to bind acetylcholine receptors on the sarcolemma of the muscle fiber cell. Here we have a clinical view. What if something happens to your acetylcholine receptors? So this is myasthenia gravis. This is an autoimmune disease. So autoimmune means kind of attack yourself. So what happens is your immune system thinks that your acetylcholine receptors are foreign, that they're not supposed to be there, and it starts attacking them and removing them. So it doesn't matter how much acetylcholine you're releasing into the synaptic cleft if you don't have the acetylcholine receptor for the chemical to bind to, you're going to have muscle weakness, decreased muscle stimulation. And at first it's just eye and facial muscles, and then eventually it's other muscles like those that help you swallow. So kind of these clinical views are for you to see that there are diseases and issues that directly relate to some occurrence within one of these 4 phases of muscle action. So it's kind of worth knowing the level of detail that we're going into with this chapter. And I promise you, when you do drug interactions, it will help you down the road if you understand all of the mechanisms and everything that's happening within these 4 phases. Now we move on to excitation-contraction coupling. This is step 2. So we saw that the neuron generated a message that caused calcium channels in the synaptic knob to open. Calcium came into the synaptic knob and caused the vesicles to release acetylcholine. The acetylcholine got released into the synaptic cleft, and the acetylcholine bound to the acetylcholine receptor. What happens when this neurotransmitter acetylcholine binds? The gates open. So the acetylcholine receptor has a gate inside, and when the acetylcholine binds, it opens. And now the sodiums, all of these little purple spheres, the sodiums can travel inside. Remember, this is our sarcolemma. This is our plasma membrane. So we have tons of sodiums on the outside, making the outside of the muscle fiber cell positive. Then we have our sarcolemma, and on the inside we have potassiums, and we have other really large negative molecules. So remember, the resting membrane potential of a muscle is about negative 90 millivolts. So what's going to happen when this acetylcholine receptor opens? The sodiums are going to travel to the inside of the muscle fiber cell. So if I have something that's negative 90 and I add 10 positive sodiums to it, so this is my resting membrane potential, it's overall negative. I add 10 positive sodiums, I now come to negative 80. I'm becoming more positive. What happens if I add 50 sodiums? I now add negative 30. So even though I'm still negative, I'm becoming more positive. So these acetylcholine receptors bind, the acetylcholine receptor opens, sodiums travel through the acetylcholine receptor, and they cause the inside to become positive, inside of the muscle fiber cell. We call this an end plate potential. We changed the inside, just right here, we changed the inside of the muscle fiber cell from negative 90 and we made it slightly more positive. So here's the wording for what we just looked at. The acetylcholine receptors are chemical gated channels. What has to bind? When I say chemical, that means a chemical has to bind to the receptor in order for it to open. So acetylcholine, we have our acetylcholine receptor, and what binds to our acetylcholine receptor? Acetylcholine. And what happens to this channel? It opens. Sodium is going to diffuse into the muscle fiber cell through the channel, and a little bit of potassium is going to diffuse out. I kind of call this like leaky, right? So a little bit of potassium is going to make its way out. The cell membrane, right, the sarcolemma, briefly becomes less negative at the end plate region. This is local. So only directly under where you have the acetylcholine receptors, this is the only area that's becoming positive. That's why they call it an end plate potential. These positives can spread once they're inside the muscle fiber cell. They can spread either way, and they can lead to other channels opening, which is what we're going to look at on the next slide. So if I extended my sarcolemma out here, I would have over here my acetylcholine receptor, and I would have my acetylcholine bound. Sodium would have come inside the muscle fiber cell, right? The inside locally became more positive. So we went from negative 90 millivolts and we became more positive. And this was called an end plate potential. It's only happening right here underneath the acetylcholine receptors. But these sodium positive ions that we've let in can diffuse. They can move from high concentration to low concentration, right? That's what diffusion means. And they can cause these other voltage-gated channels to open. So now this positive comes over here, and if the inside changes from negative 90 to slightly more positive, this gate opens. This voltage channel gate opens, and guess what happens? More sodium comes into the muscle fiber cell. So it's almost like a two-step process. The first thing you're having happen is the end plate potential. You're generating a local potential where the inside of the muscle fiber cell becomes slightly more positive. Then that positive travels over here to these voltage only, so it's sodium only channel. And now this gate opens and tons of sodium comes pouring in. So in step one at the end plate potential, we're going from negative 90 millivolts to about negative 55 millivolts. Once all of these sodium channels open, we go from negative 55 millivolts to positive 35 millivolts. So you can see there's a lot more action kind of happening here, right? So when these voltage only gates open, it's a flood of sodium that causes the muscle to become positive on the inside. And we call this an action potential. So here we're generating an action potential. The end plate potential reaches threshold, which is negative 55 millivolts, causing nearby voltage-gated sodium-only channels to open. Now we have tons of sodium diffusing into the muscle fiber cell through these voltage sodium-only channels. This causes a depolarization. The inside becomes less negative and eventually reaches this positive 30. This results in the opening of more channels, and this chain reaction occurs until the message, the depolarization, the action potential, is propagated down the T-tubule. Remember, we still have to get to the core where we have our sarcomere, because that's what's actually going to do the contraction. So what's happening over here, we have the sodium channels are opening, right? And so tons of sodiums are flooding in, and they're changing the inside of the muscle from negative 90 to positive 35, and then they're diffusing. When it reaches the sarcoplasmic reticulum, specifically the terminal cisternae, that charge difference causes these voltage gates to open. When the voltage gates open, they release calcium. And this is going to be important later because the calcium is going to bind troponin. The action potential travels down the T-tubules, causes calcium channels to open, releasing calcium. The release of calcium from the sarcoplasmic reticulum, specifically the terminal cisternae, is what's going to interact with the myofilaments and trigger contraction. So the calcium is going to interact with the troponin, and if you remember, troponin is going to remove the gate, which is tropomyosin, and then you're actually going to be able to have contraction. So just after the sodium channels open, and that's when we have our flood of sodium ions, right? And when these sodium only channels open, that's when we go to our positive 30 millivolts, right? They're going to close, and then we're going to have potassium channels open. Potassium is going to diffuse out of the cell and repolarize, meaning bringing the inside of the muscle back to negative 90. And this happens over and over again, this depolarization, repolarization. So let's look at that in more detail. Here's our sodium-only channel, and it's followed by a potassium-only channel. So over here, we generated our end plate potential. That triggers the sodium-only gate to open, and now we have a flood of sodium. And what happens to the sodium? It moves, it dissipates, it diffuses. Right after the sodium-only channel opens, the potassium-only opens, and potassium leaves the muscle fiber cell. So you're going from outside positive, inside slightly positive, right? We're at -65. Then you have the sodium gate open, and now we went from negative to +30. So now the inside is completely positive. When this potassium gate opens, what happens? We kick potassium out. Now we go back to our positive on the outside and negative on the inside. So these terms are depolarize and repolarize. So depolarization happens when you bring the sodiums inside. So let's do resting membrane potential. The outside of the muscle is positive, the inside is negative, right? Then what happens? We open the sodium only gates and sodium comes inside. So what happens? The outside is still positive, there's still tons of sodiums out there, but now we have a positive inside the muscle fiber cell. So polar meant opposite, so dipolar means both sides are positive right now. We've depolarized the muscle fiber cell membrane. Then we kick potassium out and we bring the inside back to negative. So we call this repolarization. So you should be able to look at this graph and kind of have an idea of what's happening, or play the video in your head of what's happening at each step here. So here we have our resting membrane potential, our muscle at rest at -90. Threshold is about -55, -65. What's happening to cause the inside of the muscle to go from -90 to -65? That's your end plate potential. That's when your acetylcholine binds your acetylcholine receptor, and we open up that channel and sodiums come inside the muscle fiber cell. How do we get over threshold? What's happening during depolarization? We're letting tons of sodium in. This is when the sodium only gates are open. And we have that flood of positive sodiums, and we go all the way up to +30 millivolts. Then we have repolarization. What is repolarization? When the potassium only gate opens and the inside rapidly becomes negative again, and we come back to our resting membrane potential. Okay, so let's go over the entire process again. We know that our acetylcholine has bound the acetylcholine receptor. We know that sodium is traveling through the acetylcholine receptor, and we're creating a locally positive environment inside the muscle fiber cell. That local positive is called an end plate potential. We also know that at the same time sodium's coming in, some potassium is trickling out, just a little bit. It doesn't really change. We're still, the inside of the muscle fiber cell is becoming more positive, right? So end plate potential, we've gone from -90 to -55, 65 millivolts. The sodiums are going to diffuse, right? They go from high concentration to low concentration. They're going to spread out. But that positive is going to trigger this voltage sodium only channel to open. And now you're going to have a flood of sodiums entering the muscle fiber cell. This is where you go over threshold. You go from -65 to +30 millivolts. So now the inside is depolarized. Remember, we also have the blue here, which are the potassium only. So as these ions move, they're going to trickle down here and cause more sodium gates to open. The potassium gates are going to bring the inside of the muscle back to resting membrane potential. So when the potassium only gates open, we go from positive 30 millivolts back down to our negative 90 millivolts locally, right here. What's happening to the sodium ions? They're traveling over here to the terminal cisternae, and they trigger more voltage gates to open, and that causes calcium to be released from the terminal cisternae. So now we finally get to contraction, sometimes called cross-bridge cycling. Most textbooks call this the contraction phase or step. So the calcium that you just released from the terminal cisternae binds troponin, and it causes the tropomyosin to move. Remember, the tropomyosin is the gate. Now the actin is exposed. So you have the myosin head and you have the actin molecule, and they can actually make contact now because we've unlocked the lock, right? So this is the key. We've used the key to unlock the gate and move the gate. So now the actin is exposed and the myosin can reach up and grab the actin now. Here are the steps that are going to repeat over and over again for cross-bridge cycling or contraction. So the myosin head can attach to the actin. Why? Because we unlocked the lock, which was troponin, and we moved the gate, which was tropomyosin. Now we can do something called the power stroke because the myosin head can pull on the thin myofilament towards the center of the sarcomere. Remember, we had Z disc to Z disc, and we have our thick myosin heads, thick myofilament with myosin protein, and then above it we have our thin actin filament. So the myosin heads are going to reach up, they're going to grab the thin, and they're going to pull. ATP binds the myosin head, causing it to release from the actin. Then we're going to see ATP split into ADP plus P, and that's what provides the energy to cock the myosin head. So one of the things that's hard, I'll show you when we get to the figure again, myosin kind of looks like this, right? That's not enough to reach up and touch or make contact with the actin thin myofilament. So the myosin head actually has to cock. It's kind of like flexing your forearm. The myosin head has to cock in order for it to bind to the actin myofilament. So in order to make the myosin contract, you need energy from ATP. Here we have the calcium binding. So you can see the calcium has bound the troponin and moved the tropomyosin. You can see these black kind of circles, those are the attachment points for the myosin. So basically we've uncovered the active site on the actin. So now the myosin can bind. The myosin is going to reach up and touch that active site on the actin. You can see that ADP and P are present. That means that ATP bound at some point, right? And we split the ATP, we pulled the energy out. That was to cock the myosin head. So we'll come back around to that in a second. So the myosin head reaches up and touches the actin, and you can see that the ADP and P are released, and that's what causes the pull. So the myosin head has reached up, it's touched the actin, we release the ADP and P, and that is the forward momentum to pull the thin myofilament over the thick. Then ATP is going to bind for detachment. So for the myosin head to release from the actin, you have to have ATP. ATP is going to bind, you're going to pull the energy out, and that's what causes the myosin head to cock to begin with. So the ATP binding here releases the myosin from the actin and cocks the myosin head at the same time. And then we do this entire cycle over and over again. So if you remember here, here's our Z disc to Z disc. What are we doing? We're pulling the thin myofilament over the thick myofilament. So the myosin head reaches up, it grabs, and it pulls the thin myofilament. So what happens to the sarcomere? It gets shorter, it shortens, and that is contraction. But remember how many sarcomeres there are. There are thousands of sarcomeres in a muscle fiber cell. How many muscle fiber cells are inside of a fascicle? How many fascicles are inside an entire muscle or a whole muscle? So this is happening thousands and thousands and thousands of times, and you need tons of ATP in order for this event to occur. This cross-bridge cycling will continue as long as you have calcium and as long as you have ATP. The sarcomeres are going to shorten, the Z-discs are going to move closer together. The thick and thin filaments remain the same length, they just slide past each other. This is why it's sometimes called sliding filament theory. So here's the beginning of the sarcomere, Z-disc to Z-disc, and this is what it looks like when it's fully contracted. Do you see how the thin have been pulled towards the M line, towards the center? So here's where the thin were. Now they're overlapping each other, right? So this is a fully contracted sarcomere. Here's another clinical view. There are some neurotoxins specifically from bacteria, and those toxins can cause muscle paralysis. So specifically tetanus is going to block neurotransmitters in the spinal cord, and it can actually result in overstimulation of muscles, so spasmings, right? And this can be life-threatening. Botulism, another toxin from another bacteria that prevents acetylcholine from being released at the synaptic knob. Remember, this is the beginning of excitation. If this step stops, you have no contraction. Finally, we have muscle relaxation. So the muscle has to relax so that it can do this entire process over again the next time the brain tells it to, right? So there's a termination of the nerve signal. Remember, this nerve signal was coming from the brain. This is a voluntary, skeletal muscle response. So the nerve signal has to stop. Your brain has to say, okay, stop contracting that muscle. The acetylcholine released from the neuron will stop, and the acetylcholine will be broken down by acetylcholine esterase. So hopefully we see this ending and we think enzymes. This is the enzyme that pulls the acetylcholine off and breaks it down. So if the acetylcholine is missing, the acetylcholine receptor is going to close, and you stop having an end plate potential. So then you have no more sodium-only gates opening, you have no action potential generated. That's going to close the calcium channels. The calcium is going to return to the sarcoplasmic reticulum. You're going to have the troponin, the lock, is going to become closed again, and the tropomyosin, the gate, is going to block the actin active sites again. And the muscle is going to return to its original position. So I wanted to show you real quick when I mentioned those springs, right? I mentioned this protein earlier, this titin elastic protein. You can see during contraction, because the heads have reached up and they've pulled the thin myofilament over the top, and Z-disc to Z-disc got shorter, you can see the springs are compressed right here. As soon as this link is broken, as soon as the cross-bridge link is broken and there's no more signal to contract, these springs are going to let go, and we're going to return the sarcomere to its original relaxed position. So those springs, that protein titin, which is an elastic protein, is really important for returning the muscle back to its original sarcomere length so that you can recontract again. Real quick, let's sum up this PowerPoint by talking about calcium and ATP. So we've mentioned how important ATP is, and in bones we mentioned how having the right amount of calcium in your blood is important. So remember, calcium is necessary, we've said this several times, for all muscle cell contraction and all neuron communication. So in excitation, we just learned that calcium is what triggers the release of acetylcholine from the synaptic knob. So in order to turn the neuron message into a muscle message, we have to have calcium. That's pretty important, right? And this calcium comes from extracellular. So we haven't really talked about extracellular fluid and blood, but basically it's outside of the muscle fiber cell. It is extracellular calcium that comes from your bloodstream into your interstitial fluid. So you have to have this extracellular calcium level in order for it to be triggered to enter the synaptic knob to release the acetylcholine. Then we also know that calcium binds the troponin and changes the shape so that the tropomyosin, the gate, can make the active site on the actin available. This is intracellular. This came from inside the muscle fiber cell. But how did it get there, right? We had to have it in our bloodstream in order to put it into our muscle fiber cell. And then in relaxation, we have to pull the calcium off of the tropomyosin. If we want the muscle to relax, we have to pluck the calcium off the tropomyosin. We have to put it back into our sarcoplasmic reticulum, specifically our terminal cisternae. So you can see how calcium plays a critical role in muscle contraction and neuron cell communication. We know ATP is critical in muscle contraction, but make sure you can tell me why, right? Myosin cross-bridge, the binding and the release. So the cocking of the myosin head and then the release of the myosin head, we need ATP for. In order to pump the calcium back into the terminal cisternae, it was out in the cytoplasm or sarcoplasm, right? So to pump it back against its current, remember that's active transport. If we're going from low to high, if we're jamming it back into the terminal cisternae, we're going to need ATP to do that. And the last thing I want to talk about is the sodium-potassium pump. This was another example of a protein-assisted active transport where you needed ATP for this to happen. So what were we doing in muscle contraction? We had sodium on the outside, we had potassium on the inside. The sodiums came in, and this was depolarization, or you want to say contraction, I'm okay. And then what did we do during repolarization or relaxation? We took the potassiums and we moved them back out. So now we're here. Is this how we started? No. What do we need? Sodium-potassium pump. So the sodium-potassium pump over here is going to take the potassiums and put them back to the inside and take the sodiums and put them back to the outside. We're basically resetting the playing field so that we can contract again. So without a sodium-potassium pump, you could not have ions on the right side. You wouldn't have the correct resting membrane potential. You wouldn't be able to contract again. You wouldn't be able to relax. So ATP is crucial for all of these events to occur. I want to make sure we're getting the big picture of what's happening here. So how much ATP am I talking about? How many myosin heads? How many sarcomeres? So remember, sarcomere to sarcomere, and there are several that run the length of this myofibril. And then we have tons of myofibrils inside one muscle fiber cell. And then how many muscle fiber cells do we have inside of a fascicle? And then how many fascicles make up the whole muscle? So I want you to see that these are thousands to hundreds of thousands of sarcomeres in one skeletal muscle that all need ATP and that each have several hundred myosin heads. So this is a lot of ATP that's necessary. This is a lot of contraction, and it's spreading out over the entire muscle. So if the sarcomere contracts, then the entire myofibril contracts. Then the entire muscle fiber cell contracts. Then the fascicle contracts. Then the entire muscle can contract. And you can also see why we have to wrap all of these sections in connective tissue, right? We're passing the message along from the core interior of the muscle to the entire muscle.